everybody. Some of you were in an earlier session, may recognize a few of these slides, but hopefully there will be some new ones. The usual safe harbor statement, I refer you to our SEC filings. I will be making some forward-looking statements today. I want to talk about the company a little bit, and more importantly about the technology, and focus today's talk on clinical programs in spinal cord injury and in age-related macular degeneration, in this case in the dry form, which represents the majority of patients with AMD, and a few words on investment rationale and catalysts. Not all cells are stem cells, and a neural stem cell is a cell that has some remarkable properties, and I'll explain that and become evident in a moment. But Stem Cells Inc. is the leader in neural stem cell research and development. We got one of the broadest intellectual property portfolios in the world in neural stem cell biology and related technologies, including how we culture the cells. As a company, we're first to isolate and expand human neural stem cells, which we call HUCNS-SC. We were the first to use these cells in an FDA-authorized clinical trial and to transplant these cells into the brain, spinal cord, and the eye. We are now in phase 2 clinical trials in 2 major indications, spinal cord injury and dry AMD. So these neural stem cells represent a first-in-class biologic. They're a highly purified, expandable, bankable product that remain in their undifferentiated state in the cell banks. And we have data on these cells going back 15, 16 years. The cells are stable in cryopreserved form. They only differentiate when you put them into a host. These cells do not need to be manipulated ex vivo. These are the cells, the natural cells that are unmodified, and we do not manipulate them. So we just transplant them from the cell bank directly into the patient. We have conducted clinical trials in 4 indications and 3 elements of the central nervous system, and we have not observed any cell-related safety concerns. We have human safety data from 4 completed trials involving 43 patients and adding, I might add, now that we're in phase 2. And the longest follow-up data we have on patients is over 5 years. We've demonstrated preliminary efficacy, and we'll talk about that in a moment. And this early data from the phase 1-2 studies replicates the very exciting data that we've published in the animal studies. And very importantly, as I said, not all cells are stem cells. These are acting like stem cells in that not only do they engraft and migrate into the host, within the host tissue, but also the cells survive for many, many years post-transplant. We have evidence that they survive at least 6 years, and there are no immune response issues when we withdraw immunosuppression, and we've data that shows the cells are viable and present at least 5 years post withdrawal of the immunosuppression. This is a very brief overview of the clinical program. We've got spinal cord injury, which I've talked about, retina, the age-related macular degeneration is the current focus, and neurodegenerative diseases, lysosomal storage disease, and white matter tract disorders. The 2 studies that are in phase 2 now involve cervical spinal cord injury and dry AMD, and I'm going to focus the rest of my comments on that for the rest of the talk. This is a gentleman who was one of the earlier enrollees in our thoracic spinal cord injury study, and I will leave the quote to speak for itself. He's doing extremely well, and we're very proud of him. He truly is a pioneer. So let's talk about spinal cord injury. What is the prevalence? Well, there are approximately 1.3 million patients in the US. About 40,000 cases per 1 million population per year, which amounts to about 12,000 cases a year in the US, and about double that amount in Europe. Approximately half of these patients have complete or incomplete tetraplegia. It's a very expensive piece of business for the US healthcare system, because typically the victims of spinal cord injury, if they survive the traumatic event, usually a car, motorbike, or sports injury, they have a long life to look forward to. And so on average, this can amount to $1.5 to $4.5 million direct cost to the healthcare system over the life of the patient, if you start counting from age 25 on. Also interesting, there's a very low rate of return back into the employment group, with 11.8% 1 year post-injury. And there are no treatments available that have been proven to restore spinal cord injury. There have been various attempts, but none of them have panned out. So the phase 1/2 study design was a study focusing on the thoracic area of the spinal cord. This is the paradigm. You start lower in the cord, and then you move up to the cervical region, which directly controls nerve function of the upper extremities and motor function. This was an open label, single dose, 9 months of immunosuppression. The study was approved by Swissmedic, Health Canada, and the FDA. The primary objective, of course, was safety. The secondary objective, of course, was to look for evidence that these cells were doing something useful in the patient population. So we were using light touch and reaction to pinprick, temperature, and electrical stimulation. We used electrophysiological techniques, SSAP, and heat potentials. We also followed the patients for quality of life. Enrollment and transplantation was completed in April of last year, and we reported the top-line results in May of this year. Phase 1/2 study, small number of patients, open label, all of the appropriate cautions. But what was interesting to note was that 7 of the 12 patients in the subject reported gains in sensory function. And remember that these were transplanted into the thoracic area of the cord, and these are predominantly sensory neurons at work in the thoracic region. 3 of the subjects of the 7 were Asia A's, and 4 of them were Asia B's. So we also noticed that approximately 3 months after the transplantation, we started to see effects that these cells were doing something useful in sensory function, and also that the regained sensory function was maintained. It wasn't transient. We also noticed that the less severe injuries gained most pronounced results. In other words, the Asia B characterized patients did a little bit better than the Asia A's. And we used electrical and heat sensation thresholds to confirm changes in pinprick and light touch, because, of course, the patient can see the physician coming at them with a pin or a Q-tip. And so we use these more objective measures to confirm their response to the pinprick and light touch. What was also very interesting and unexpected was that 2 of the subjects converted from complete to incomplete spinal cord injury, and their classification was improved from Asia A to Asia B. So in summary, this was the first ever neural stem cell transplant in spinal cord injury. It was the first clinical trial to use quantitative sensory testing. The safety, both above and below the level of injury, was a very, very good benign safety profile. And I've already mentioned it was supported by sensitive and extensive testing, clinical and neurophysiological concordance, and a safe injection into the intact cord above and below the level of injury. Preliminary efficacy, as I mentioned, improved sensory gains, definitely showing biological effect of the cells, and provided us with the rationale to advance to the phase 2 study in cervical spinal cord injury. So now we're moving up above the cord, we're moving up higher in the cord. This study is called the Pathway Study. Our Chief Medical Officer, Stephen Hune, who's responsible for the clinical activities of the company, has stated that the corollary of the sensory improvements that we observed in the phase 1/2 thoracic trial could well be improved motor function in the upper extremities of patients with cervical spinal cord injuries. And so this is very exciting because while everybody is interested and concerned about the well-being and the care of these patients, sensory function is not something that really catches people's attention, particularly in the investor community. The phase 2 study, which I want to focus on for a moment, explain the design. This is designed to establish clinical proof of concept. It's a randomized study, controlled, single-blinded. This study has been approved by the FDA and Health Canada. Will involve a total of 52 subjects with injuries in the C5 to C7 area of the cervical spinal cord. Asia A's will be enrolled 4 months to 12 months post-injury, and Asia B's from 4 to 24 months post-injury. So we're certainly outside of the area of spontaneous recovery, and there is a fair degree of spontaneous recovery in the earlier phases of injury to the cord, assuming the patient survives the original insult. The safety and preliminary efficacy endpoints include dose confirmation, changes to INSCI upper extremity motor score, which is a well-established, tried and true system to evaluate motor score changes. And we'll also be looking for changes in lower extremity motor score, motor level, and sensory scores. We'll follow these patients for 12 months post-transplant. To date, we have 12 sites actively enrolling. There are more to follow. We completed enrollment in an open label study involving 6 patients in April of this year. We commenced dosing in our enrollment, I should say, in cohort 2 in June, and we are continuing at a steady pace, and our goal is to complete enrollment in 2016. Because the first cohort was open label, we will be able to share the data from that first cohort, and this will be the first look at evaluating the potential for cells to improve motor function in the cervical region. And we will be releasing data in Q4 of this year. This will be data that will be assessed, the 6-month data for all 6 subjects who entered that cohort. 12 months data will be available in Q2 of next year, and then top-line data for all 3 cohorts will be available in 2017. I want to move now on to dry age-related macular degeneration. Again, this gentleman was one of the first entrants into the phase 1/2 study, and he also is doing very well. Age-related macular degeneration is the #1 cause of vision loss and legal blindness in people over age 55. Prevalence in the US, a large number of people, 10 million, with estimates of 25 to 30 million worldwide. There are classically, people refer to the wet form of AMD and the dry form. The wet form represents 10 to 15% of all cases, and as you know, there are several treatments available, but again, no known cure. In the dry form of AMD, which represents up to 90% of the cases, there are no approved treatments. And the geographic atrophy form represents about 15% of the cases. This is the most advanced form of dry age-related macular degeneration. We did a phase 1/2 study, and we announced the results of that study in June of this year in Stockholm at the annual meeting of the International Society for Stem Cell Research. We observed that BCVA, or best corrected visual acuity, was stable or improved. We also noted gains in contrast sensitivity, which is a functional measurement of the ability of patients to identify between white and black and dark and bright. We also observed a number of anatomic changes, increased macular volume and foveal thickness in the study eye. This design was such that we were able to use the less affected eye as an inbuilt control. And so this is evidence or a suggestion here that we're improving the health, if you will, of the macula. We assessed geographic atrophy and the rate of progression of geographic atrophy using fundus autofluorescence, or FAF. And it's interesting to note that there were 15 subjects in this study, and when we looked at the data for the phase 2 study, we were able to identify 1/3 of the patients who would meet the eligibility criteria for the phase 2 study that was on the drawing board. The major area of interest in terms of eligibility is lesion size. If the lesion is way too large, it's very, very difficult to measure millimeters of change in a relatively short period of time. And similarly, if it's too small, you have a similar problem of measurement. But we're very interested in how well these patients did in the phase 1/2 who qualify for the phase 2 study had they been enrolled in that study. Now, the results I'm going to show you for these 5 patients, these results were confirmed by 2 independent reading centers. So essentially, if you look at this chart, the study eye on the left and the fellow eye on the right, and the red line, or 45-degree angle, represents no difference or no change between the control or the study eye. So if you have a measurement which is to the right to the right and lower of that red line, then essentially what it's saying is that the fellow eye is advancing more rapidly in terms of degeneration of the macula, is advancing more rapidly than the controlled fellow eye. So this is very encouraging, and it suggests to us that with the target patient population for the phase 2 study, we're definitely on the right track, and we appear to be having an impact on those studies with lesion sizes who will be enrolled into the phase 2 study. So just in summary, we've shown that subretinal transplantation of cells is safe and well tolerated. We've seen preliminary efficacy, and we've seen significant anatomic changes. And the phase 1/2 outcomes that I've shared with you support further clinical development. So the phase 2 study is called the RADIANT study. Again, this is designed to establish proof of concept. It's fellow eye controlled, and the thinking is to enroll 63 patients in 20 US sites that will be expected to participate. This study got up and running a little bit later than the Pathway study, so we haven't made as much progress yet in terms of number of sites, but we have dosed the first patients. The preliminary efficacy endpoints, the primary anatomical measurement will be focusing on measurement of the rate of increase of geographic atrophy of the treated eye compared to the fellow eye. And then secondarily, we'll be looking through OCT, BCVA, luminescence, BCVA, contrast sensitivity, and a host of other measurements for each of these patients. And again, like the phase 1/2 study, we will follow these patients for 12 months. The significance for this phase 2 study, I think, should be pretty obvious. If these cells prove effective for photoreceptor protection, there clearly is a growing need. The incidence of AMD is expected to increase dramatically, tripling by 2025 as baby boomers continue to age. It may also hold promise for treating other retinal degenerative diseases, such as retinitis pigmentosa, which is the most common inherited cause of blindness that affects an estimated 1.5 million people worldwide. So I'd now like to just finish up with a couple of slides talking about investment rationale and introduce you to the management team. Clearly, there are large unmet market opportunities. We have a proprietary technology, the potential for a one-time intervention to treat numerous CNS disorders with significant unmet need and no approved therapies. Proof of concept studies are well underway, and the phase 1/2 data is encouraging and consistent with preclinical data, demonstrating a favorable safety profile and signs of efficacy. In the middle of this year, we did a capital raise. We raised over $25 million to strengthen our balance sheet. I believe there's a compelling current valuation attached to this, to Stem Cells Inc. that has a buy rating from 5 analysts who follow the company. Catalysts in the near term, we will have significant clinical milestones in Q4, and we're on target for that. We will be releasing incremental data points in 2016 and top-line phase 2 results in 2017. And last but by no means least, often overlooked and forgotten, are the people who are really in charge of this technology and working it through the very arduous development programs that you need to undertake for success. I've been in this business for more years than I'm prepared to admit in public, but I guess I have to here. I got to fess up. I've been in this industry for over 40 years. I've done a couple of life sentences at BD and Abbott and BOC Healthcare. Our president and chief operating officer, Ian Massey, 30-plus years in the pharmaceutical industry with a small, relatively small pharma company called Biotech, Sanofi, Syntex, and Roche. Greg Schiffman, our chief financial officer, who's here with me today, has over 15 years life science and biotech experience with Affymetrix, Applied Biosystems, and yes, Dendreon, which is the subject of a previous panel discussion. Stephen Hune, he's a pediatric neurosurgeon. Prior to joining Stem Cells in 2007, he was chief of pediatric neurosurgery at Stanford School of Medicine. Dr. Ann Tsukamoto is our EVP of Scientific and Strategic Alliances. She joined Stem Cells Inc. in 1998 as the first name on the human hematopoietic stem cell, and she led the charge to discover the human neural stem cell at the company. Very, very experienced and talented researcher. And one of the newer members of the team, Dr. Joel Naor, an MD ophthalmologist. He's the VP of Clinical Development for Ophthalmology with over 15 years in the field with Santen, Macular, Allergan, and QLT. So I thank you very much for being here and for your attention.